Hi everyone, so today we get a chance to talk about biomolecules again. Okay, this is our video 3 out of our 4 series of 4, and today we're going to talk about nucleic acids. Okay, what is the function of nucleic acids? As it says, well, actually, they're located in the nucleus. Okay, and they are in charge of storing genetic information, DNA, RNA, etc. Okay, so for this, okay, biomolecule, we're going to focus on the nucleotide, okay, which is the monomer, the repeating structure, and we're going to take a look at, okay, one example of a nucleic acid. We're going to learn how to decipher this in a few minutes. So to start, okay, we have the nucleotide. The nucleotide is the repeating structure. It's a combo of 3. Like when you go to the store, you get a combo, you get soda, you get a fries, and you get a burger. Okay, you get 3 things. In this case, you get your phosphate, you get a sugar, and you get your base, a nitrogenous base. So phosphate, carbohydrate, and nitrogenous base. So those are the 3 parts of the monomer. So the phosphate, okay, it's a phosphorus group with 4 oxygens around and a negative. So PO4 minus. That is what a phosphate is, okay? It's a common repeating structure in biology. Then we have our carbohydrate. If you check video number 2, you will see that we have these structures like hexagons and pentagons repeating over and over again. What is characteristic is that they have hydroxy groups on every carbon, okay? And finally, nitrogenous base with nitrogen in their basic, okay? So we're gonna make we're gonna talk about, okay, what are the different kinds we will encounter in nucleic acids of each of these 3 parts. So to start, we have the phosphate group, okay? This is the same for both DNA and RNA, okay? So then we move on to the carbohydrate. Here we actually have 2 choices. We have the one in DNA, deoxyribose, and the one in RNA. What's going to be their difference? So in DNA and RNA, they look about identical, almost the same. So you notice here, it's always something like this. Now, the only change that you will see, or the way you can identify DNA from RNA by looking at the molecules that make up the backbone, which would be the phosphate and the sugar, will be by looking at the sugar and checking this, okay, carbon number 2. Okay, in DNA, D stands for deoxy. Okay, all right, deoxyribonucleic acid. So that means this is a hydrogen by itself. Okay, ribonucleic acid. That means this ribose, okay, is completely oxygenated. Sorry. So the difference between RNA and DNA is going to be this oxygen. Okay, with respect to the carbohydrates. Next, we have our nitrogenous bases. So what are those about? Nitrogenous bases, okay, is this 3rd group that we have over here. Okay, you have lots of nitrogen, so far the only one. And you will have 2 groups. We have the purines and we have pyrimidines. So what's the difference among these 2? How can you identify them? Okay, you will see that purines and pyrimidines are bases that have either 1 ring or 2 rings. So for example, okay, sorry for this. Okay, purines are going to have 2 rings. All right, so here we have an example. Alrighty. Okay, you see 2 rings, and pyrimidines are going to have only 1 ring. Okay, so let me show you a few examples of these 2. Okay, so for example, here we have adenine and guanine, and if you notice here, you have 1 ring, 2 rings. So this is a purine. Okay, 1 ring and 2 rings. So it is also a purine. So adenine, you've probably seen it as A before, and guanine as G. Okay. Pyrimidines, on the other hand, well, they are single rings. So if you notice here, 1 ring, one ring, one ring. Why do we have 3 instead of 6 or 4 only? Well, because uracil is present in RNA only, okay? And thymine is present in DNA only, okay? And they are examples of pyrimidines. Okay, so those are our different possibilities, okay? So now, let's go ahead and take a look at the major leagues, all right? Let's go ahead and take a look at a piece of DNA, okay? So when given a piece of DNA, you can be asked a few things. So for example, to identify a phosphate group, we talked about it, okay? We said that, well, this nucleotide was the repeating structure. So if you remember this nucleotide, okay? It's like if you repeat it over and over and over and over and over again. So you have phosphate, carbohydrate, and nitrogenous base. Phosphate, carbohydrate, nitrogenous base. So to identify a phosphate, for example, here you have the phosphate in red, okay? So here you have another one, here you have another one, okay, etc., etc. All of these are phosphate groups, this, okay? And this one as well. Now, next is to identify your carbohydrate. So what would be the carbohydrate? The carbohydrate or the sugar will be, okay, precisely your sugar. So it would be this, okay, ring that has the oxygen in here. So this is another one. In this case, is this deoxyribose or ribose? You would look at here, has no oxy, so that is DNA, deoxyribose. Okay, finally, we have to identify our nitrogenous base. So, and if we see one ring, okay, that means we're talking about a pyrimidine. Okay, so this is a pyrimidine. If you're talking with 2 rings, if you're asked to identify 2 rings, that means we have a purine. Alrighty, so this is a pyrimidine. So these 2 chains are joined, okay, by hydrogen bonds. Hydrogen bonds are intermolecular forces that occur among molecules, and these are the hydrogen bonds. Okay, you have 2 among thymine and adenine, and you have 3 among cytosine and guanine. Okay, now what if you were asked to find or to identify one nucleotide? What would you signal? Well, remember, a nucleotide is a combination of phosphate, sugar, and base. So you would have to circle everything that is a phosphate, a sugar, and a base. Okay, then another question could be, for example, to identify a codon. So the code, 3 nucleotides have very important information for protein making, which will come on the next video. But to know and recognize that every 3, okay, of them is going to have this key information. So 1, 2, 3. So you show that 3 of them, okay, not all the way to the end here, okay, but just through the center. Remember, these are intermolecular, so they are not really bonds. This is a codon, so this code would be, for example, GAG. Okay, so that will be it. Hope this worked. Okay, don't forget you have a nucleotide, DNA versus RNA is an oxygen. Okay, nitrogenous bases could be purines or pyrimidines, and to identify the different parts when you have quite a big molecule, okay, phosphate, sugar, and base, hydrogen bonds, okay, pyrimidine, purine, nucleotide, and codon. Okay, good luck.